All right, now for the other part of the notes, mitosis. So these are the actual steps in the steps of mitosis, the division of the cell. So interphase, that's the G1S, G2, okay, where the cell spends most of its life over here in interphase. That's what this is. In that state, the DNA is all loose and we call that chromatin. It's a chromatin state. Okay, so a few things visually to look at here. We have here the membrane around the nucleus. Okay, that nuclear membrane in interphase. In prophase is both early and late stages. In prophase, that nuclear membrane dissolves. So here you can see that nuclear membrane is starting to fall apart. Okay, begins to dissolve in the beginning, and then by late prophase, it's pretty much gone. Okay, now what you're looking at here inside the cell, okay, the nuclear membrane disappears. This loose chromatin, which is the DNA is all loose, it becomes condensed into chromosomes. So these are visible chromosomes. Right? They're condensed visible chromosomes. So the DNA condenses, the chromosomes are becoming visible. All right, chromosomes in mitosis, you're going to see them paired up like this. Okay, you have a chromosome that has been replicated. Okay, so we have two identical copies of the same chromosome. Those are called sister chromatids. So this is one chromatid, this is another chromatid, these are sister chromosomes. They're attached together, all right, at this thing right here, which is a protein that is called the centromere. Okay, put a little note here, these are replicated chromosomes. And what you see happening here in prophase, we have these little things right here. These are the centrioles, and they produce these long little spindle fibers that attach at the centromere and grab onto those chromosomes. Okay, and move them around. So that's what we're looking at down here. So kind of a visual on that. We have here okay, this is the centriole. And out of that centriole grows the spindle fibers. Okay, and those spindle fibers will go out and grab on, each one of them will grab, well, not each one, but they'll grab onto a centromere. Okay, and there'll be one of these centrioles with its spindle fibers on each end of the cell. So here we have the spindle fibers from this centriole right here, and here's the other centriole with its spindle fibers. And they are grabbing onto the chromosomes, which you are seeing here in the middle, the chromosomes. Now, metaphase is the one that's most easy to identify because the M for metaphase means all the chromosomes are in the middle. The M for metaphase is the M for middle. All of the chromosomes are lined up in the middle or in the equator of the cell. Okay? Chromosomes are lined in the middle of the cell. All right, so those are all the chromosomes. All right, and our spindle fibers, I just write that word down here. Spindle fibers. All right. Anaphase, those chromosome pairs, so each chromosome with its sister chromatid, those will be separated, the centromere will break, and those will separate. So one copy will go one direction, one copy will go the other direction. So in this case, one copy will go up to this centromere, and the other copy will go down to this centriole, and this one will go to the other centriole. So those copies, so these two identical copies separate to opposite ends. These two identical copies separate to opposite ends. These two identical copies separate to opposite ends, and these two identical copies separate to opposite ends of the cell. Okay? And the spindle fiber, that happens by the spindle fibers getting shorter and shorter and shorter. That's anaphase. Telophase, all right, what you're seeing, this is an example of a plant cell. The most important thing about telophase is the nuclear membrane is beginning to form. So you start seeing a nucleus beginning to form right here. There's one, and then another one beginning to form over here. That's two. What we started with was one nucleus, and what we're going to end up with is two nuclei. Okay? With the same identical DNA, because these chromosomes went one direction, their copies went the other. So we have identical copies on both ends, identical copies from here and here. So we have those two nuclei have the same exact DNA inside there. So in telophase, the nuclear membrane starts to reform. Once that nuclear membrane has reformed, the DNA decondenses, goes back to that chromatin state like it was in interphase. It goes to that chromatin state. And the rest of it, which is the cytoplasm, the liquid of the cells, begins to separate, and all the organelles that are in there begin to separate. And this part of the cell starts to get shrunk smaller and smaller and smaller, gets squeezed by what's called contractile fibers until it separates the two cells. Okay, so that is your notes on mitosis.